comes science. They might be giants, here comes science.
Hey, Dr. H here. This video will be going over Unit 3 of AP Biology. That unit is all about energy. So in part 1, we will go over some basics of thermodynamics and enzyme function. And then in part 2, we will get into some of the energy capturing pathways of photosynthesis. And then we'll end up in part 3 with the energy releasing pathways of cellular respiration. I think this might be a little bit longer video because of all the topics we need to get through. So I will definitely have a bunch of time codes down below so that you can find specific things that you are looking for. All right, so we'll start off with some basic facts about energy, especially a couple of the laws of thermodynamics. And these are basic laws of the universe, the way that energy converted and transferred. And when we're talking about biology, there's really two fundamental laws that we need to worry about. The first one, the first law of thermodynamics, there states that energy is neither created nor destroyed. It can only be converted from one form to another. Okay, so the overall amount of energy in any given system is constant. But in the case here of the bear eating the fish, right, that chemical energy in the food is being converted into the energy of movement, right? Kinetic energy, giving the bear the energy to run around. The second law of thermodynamics is about these conversions of energy, and anytime there is an energy conversion or energy transfer, right, converting energy from one form to another, or transferring energy from one place to another, there is always a little bit of loss, right? It's never 100% efficient. And this loss generally goes into the environment as heat, and this will increase the entropy of the universe, right? And entropy can sort of be thought of on a very simple level as the randomness of the system, right? How much disorder, right? Things tend to become more and more disordered over time, right? And that is entropy. The other aspect of energy that we need to look at is how energy relates to chemical reactions, right? How much energy is present in the chemicals at the beginning of a reaction versus how much is in the products that are present at the end of a reaction. If the products are at a lower energy level than the reactants, then that is referred to as an exergonic reaction, right? And we say that energy is released, right? And there is this measurement, delta G, that measures the change in energy. So in this case, for this exergonic reaction where energy is released, we would say that delta G is negative, and that the products are at a lower energy state. And this energy, of course, it has to go somewhere, so it is released out into the environment. Now, the opposite of that would be an endergonic reaction. And this is a reaction that would require the input of energy. Right? And as we see on the graph there, the products are at a higher energy level than the reactants. And we would say that delta G is positive. Okay, these reactions are also referred to as non-spontaneous because since they require that input of energy, they don't happen very often. Okay, the spontaneous exergonic reactions are much more likely to happen in a reasonable amount of time. Okay, and we'll certainly talk when we get into enzymes, we'll talk about how cells deal with these two different types of reactions and how they can actually allow endergonic reactions to occur at a time scale that allows living things to survive. So when we look at these chemical reactions in a cell, they are generally they are generally in a metabolic pathway, right? A series of reactions, and there are a few reasons for putting these multi-step metabolic pathways together. One of the main ones is to maximize the amount of energy that can be captured. Right? If it's just a one-step reaction and all the energy is released at once, the cell will have a very hard time capturing the maximum amount of energy from that reaction. Instead, if the reaction proceeds one step at a time, then each of those small energy releases can actually be harvested by the cell and converted into some useful form of energy at a much higher efficiency than if it's just one reaction that releases all the energy at once. The other major reason for cells to have these multi-step metabolic pathways is for regulation. Right? Whenever there are multiple steps, each of these steps is going to be catalyzed by an enzyme, and enzymes, as we'll learn in a little bit, are highly regulated. And one of the very common regulatory mechanisms that we see is this feedback inhibition. Right? This is a negative feedback loop where a metabolic pathway will be turned off by its final product. So in our example here, we have this multi-step pathway, right, converting one amino acid into a different amino acid, right? Very common metabolic pathway. The feedback inhibition is when that final product starts to build up in the cell, it will bind to that first enzyme in the pathway and inhibit it, right? It will shut down that enzyme activity. This will prevent the cell from wasting all of these resources to make a final product when the cell already has enough of that final product present. Okay, so it has evolved to conserve materials and to conserve a little bit of energy in running all of those steps when the cell does not need to generate any more of this final product. And we will see these feedback loops over and over again as we go through the rest of the content. Couple last things here about cellular energy to remember. Of course, the primary energy molecule for cells is ATP, right? Adenosine triphosphate, and that the structure here. But the reaction that releases the energy is the hydrolysis of ATP. Right? One of the phosphate groups there is removed, and that is a highly exergonic reaction. Right? It releases a large amount of energy into the environment. Okay, so cells can harvest this energy to run other endergonic chemical reactions, right? Chemical reactions that require an input of chemical energy. And the way this is done is through coupled reactions, right? Running two reactions together, one endergonic, one exergonic, and the overall energy change is the sum of the individual energy changes. All right, so let's take a look at an example of that. So at the top there, that is a fairly common chemical reaction, right? It's another amino acid conversion, right? And we see the delta G there is positive, right? So that would require the input of energy to proceed. The coupled reaction, right? The ATP losing the phosphate group and becoming ADP, adenosine diphosphate, that, remember, has a very high negative delta G. So when we look at this coupled reaction there, the overall reaction is negative, The delta G is negative, so overall this is an exergonic reaction, and it can proceed spontaneously at a rate that can support cellular life. Right? So this idea of coupled reactions is very important in understanding the way that cells use ATP. Right? They use this hydrolysis of ATP, this highly exergonic reaction, to drive endergonic reactions that require the input of energy. And when the two reactions are occurring together in a coupled reaction, then the overall delta G is simply the sum of the two individual delta Gs. So moving on from just basic energy, I want to talk a little bit about enzymes and enzyme functions. Another way to look at energy graphs of a chemical reaction, instead of looking at the bar graphs that we looked at before, we can draw these sort of line graphs here that basically show the same information, right? It is the course of the reaction on the x-axis and the amount of energy in the y-axis, right? But here we're kind of looking at the energy as the reaction proceeds. And if we look at that black line there, right, that is the course of the reaction without an enzyme, right? So even though this overall reaction is exergonic, right, the products are at a lower energy level, there is this input of energy at the beginning that needs to occur for the reaction to get started, right? And we call that the energy of activation. Okay, and this is why reactions, even though we say that these exergonic reactions are spontaneous, they may occur at very, very low rates because of this large energy of activation. So enzymes will function by lowering that energy of activation. Right? Looking at the red line on the graph there, you see that overall, many aspects of the reaction have not changed. Right? The reactants are at the same starting energy level. The products are at the same final energy level. So overall, the delta G has not changed. But what has changed with addition of the enzyme is that energy of activation. Right? It takes less energy to get the reaction started. So this is the way enzymes and really all catalysts work. They make the energy of activation lower, so the reaction is more likely to proceed with that amount of energy found in the environment at the time. So looking at this a little bit closer and trying to figure out, well, what actually is this energy of activation, right? What is happening at the top of that energy hill there? In terms of chemical reactions, that state is called the transition state, right? And this is a highly unstable state. Right? So looking at an example reaction here, if we imagine that we have two atoms bound together, right, in two molecules, right, and then the products, the atoms are bound together differently. At that transition state, what is happening is the chemical bonds are sort of in an in-between state. Right, they are half broken and sort of half formed. You can imagine this is a very unstable position for these chemical bonds to be in. So that's why it is at such a high energy level. Enzymes will function, enzymes will lower the energy of activation basically by stabilizing that transition state. Okay, so when we look at enzyme function, and enzyme structure, right? So remember, structure and function always go hand in hand. Most enzymes are proteins, and they will have a structure that allows the substrates, right, the starting materials, to fit in perfectly to this little pocket of the protein structure, right? And that is called the active site. And in that active site, the amino acids of the enzyme will be making contact with the substrate and stabilizing that transition state, right? Whatever kind of contacts they need to be, right? Whether they're ionic or hydrogen bonds, hydrophobic bonds, whatever it has to be in there will stabilize that transition state. So the sort of general cycle here of enzyme activity. Substrates come in to the enzyme active site. The chemical reaction occurs, right? It will be begun by some of the amino acids in that active site, and they also will stabilize that transition state, and that will speed up the reaction, or technically it makes the reaction more likely to occur, and now the substrates have been converted into products. Okay, and since these products don't fit as well in the active site as the substrates did, they are released, and now the enzyme goes back to its beginning state and is unchanged and is ready to catalyze another reaction. Okay, enzymes can work over and over and over again, catalyzing hundreds or thousands of reactions. Okay, as long as there are substrates around, the enzymes are going to be active and catalyzing these reactions. The last important point about enzymes is the effect of environmental factors on enzyme activity. Right? Enzymes can be very sensitive to the environment in terms of how active they are. So a couple things that can drastically affect enzyme activity. First, temperature and pH. Right? We see the graphs there of a few different enzymes. Every enzyme is going to have a optimal temperature. Right? Human enzymes there generally will be 37 degrees Celsius because that's our internal body temperature. Right? Contrast that with the other enzyme there, a enzyme that is from a heat-tolerant bacteria has a much, much higher optimal temperature, right? 75 degrees Celsius, right? Our enzymes, remember, they're proteins, and remember what happens to proteins when they get too hot, they would denature, right? These heat-tolerant bacteria have evolved heat-resistant proteins, so they do not denature and retain their activity at this very high temperature. We see very similar effects with pH. Right? There are optimal pH values or optimal pH levels for each of these enzymes. Right? And here we're looking at two different human enzymes. Right? One that works in the stomach, it's going to be very acidic, and then one that works in the intestine, that's going to be very alkaline, right? Very basic. Something else that can affect enzyme activity is the presence of inhibitors. Right, and we sort of talked about this when we introduced the feedback inhibition. Right, an inhibitor is some other molecule that's going to bind to the enzyme, bind to the protein, and lessen its activity, right, or completely remove its activity. Right, and depending on where that inhibitor binds, it will be classified either as a competitive inhibitor or a non-competitive inhibitor. Right? A competitive enzyme inhibitor is going to bind in the active site and prevent the substrate from binding. Right? And that is how it will block enzyme activity. A non-competitive inhibitor is going to bind somewhere else on the protein molecule. And by binding to the molecule, it's going to change the shape of the active site and prevent the substrate from binding. Right? Either way, the effect of either of these inhibitors is turning off enzyme activity. Right? And this is one of the major regulatory mechanisms for all of these metabolic pathways. Right? This the presence of inhibitors, and that is how the feedback inhibition works. So the first set of cellular reactions that I will go over here are the energy capturing reactions of photosynthesis. Okay, since these are energy capturing, that means that the end products will be at a higher energy level than the starting reactants. These are all then endergonic reactions, right? There is an input of energy, right? And we will see that all throughout the two different sets of reactions that we will talk about the light-dependent reactions, and then the light-independent reactions. Okay, before we get into the reactions themselves, I want to go over some of the basic structures that are responsible for photosynthesis. So first, before we really get into some of these structures, I do just want to take a second to remind everyone that photosynthesis is not unique to plants, right? There are tons of other organisms out there that can also perform photosynthesis, right? And you see some of the examples here, right? Algae, some of the green bacteria, there are protists, right? Some of the unicellular eukaryotes, and the processes that they will go through will be very similar to what we talk about here. Certainly, I am going to go over specifically plant photosynthesis. Many of the processes that plants go through will be the same or very similar to what these other organisms go through. So looking at some plant structures, certainly the main site of photosynthesis in a plant is, of course, the leaves. Looking here at a cross-section of a leaf, looking inside, we have that mesophyll layer. The bottom layer there, especially important for photosynthesis, that is the spongy mesophyll, with all of the air spaces between the cells. Right? And as we'll see, it's very important that all of these photosynthetic cells are in contact with the atmosphere, with the carbon dioxide that is coming in and the oxygen then that is being released as the byproduct. Looking a little bit deeper into the cell at the organelles involved in photosynthesis, and of course we have the chloroplast. Real quick with chloroplast structure, there is a double membrane around the outside, right? Outer membrane and then the inner membrane. Those don't play too big of a role here in the photosynthetic process, but certainly all of those thylakoid membranes inside play a very large role. And the reason for all of those membranes inside is for all of that surface area. Right, and that will become apparent why that is important when we get into the process that occurs there on the thylakoids. Okay, and everything that surrounds the thylakoids inside the chloroplast, that is referred to as the stroma. Okay, and as we move through the reactions, it's very important to remember where these different sets of reactions are taking place inside of the chloroplast. All right, and so here we see those different reactions, right? The light-dependent reactions, or just the light reactions, as on this diagram, those are occurring on the thylakoids, and then the dark reactions, or the Calvin cycle, are occurring outside in the stroma, right, outside of the thylakoids. And we see, you can see the general inputs and outputs there, which we will definitely get into as we go over these reactions step by step. All right, so the first set of reactions that I will go over here are the light-dependent reactions. And as their name implies, these require the input of light, right? That is the form of energy that is coming in to this set of reactions. As I go over all of these metabolic pathways, the most important things to keep in mind, you certainly don't need to know all of the details about molecules and structures and enzymes. The most important things are location, inputs, and outputs, right? Where does this metabolic pathway take place? What is feeding into it, right? What are the starting materials? And then what are the end products? And looking at that, breaking it down like that for the light reactions, we see that the location, as I said previously, is on the thylakoid membranes. And the major inputs there, light energy, right? Obviously coming from the sun, and water, right? Water coming from the environment of the plant. And there are three major outputs here of the light reactions. The first two, ATP and NADPH, those are the high energy products, right? And we will see those in the next set of reactions, right? They are carrying the energy that's captured from the light into the next round. And then finally, we have oxygen gas. And in terms of photosynthesis, that is considered a waste product, and that is released out of the leaves out into the atmosphere. So looking at that in terms of structure, there we have the thylakoids with light energy coming in from the sun, water coming in through the plant, right up through the roots, and our two high energy products there being released, ATP, NADPH, right, ready to go on to the next set of reactions, and water there being released out into the atmosphere as a waste product. Before I get into the actual reactions of these, before I get into the actual steps of the light reactions, I do want to take a minute and just talk a little bit about light, and a little and how it interacts with structures like chloroplasts and leaves. First off, certainly we should remember that light is just one small portion of this larger electromagnetic spectrum. We see the different wavelengths there going from purple, the very short wavelengths, up to the longer wavelengths of red. And when light hits an object, light can either be absorbed or reflected, right, or transmitted. And when we're looking at something like a chloroplast, a lot of the green wavelengths are reflected back, right? And that is why most plants, most leaves, appear green, because the green light is reflected back and most of the other wavelengths are absorbed. So when these other wavelengths are absorbed by the chloroplast, that means that it is able to capture the energy in that light. So if we look at the ability of different lights to support photosynthesis, not surprisingly, the wavelengths that are absorbed, meaning the purples, the blues, the reds, and the oranges, work very well for photosynthesis. The greens, yellows, not so much, right? Because most of that light is reflected and that energy is not being absorbed. All right, so what happens when this light energy is absorbed? Okay, so looking at the first set of reactions here are light-dependent reactions. Right, remember that this is all occurring inside the chloroplast on those thylakoid membranes. There are two large structures here that contain the primary pigment, right? The chlorophyll, right? And those are the two photosystems here, right? PS2, which actually comes first, and then PS1. So when the energy from the light is absorbed by the chlorophyll molecule inside PS2, that energy is passed along into two electrons. Okay, and those electrons then move to a higher energy state and are now these high energy electrons. Okay, and those high energy electrons are then passed on to this series of proteins embedded in the thylakoid membrane called the electron transport chain. Okay, and it's not terribly important that you know all the details of these proteins or the names of these proteins. Right? Just understand that they accept these electrons at a high energy level and then pass them along, and that energy from the electron is captured in the form of ATP. Okay, and we'll talk about that exact mechanism in a minute. When those electrons reach the end of the electron transport chain, they are then passed on to the next photosystem, PS1, in a low energy state. In PS1, there are again chlorophyll molecules which can accept light energy and excite 2 more electrons, right? And those electrons from PS1 are passed on to a very short electron transport chain, which doesn't really take the energy out of the electrons. Instead, it uses those high energy electrons to make the other high energy product of the first set of reactions, the NADPH. Okay, so here we have our 2 high energy products, right? ATP made from the electron transport chain there, and NADPH made at the very end with this enzyme NADP plus reductase, right? Which the name of that enzyme may come up. I don't know. It's always hard to say what details are going to be on the test. The one more reaction that is very important, going all the way back to PS2. Remember that those electrons were excited and then they left, right? So in order to replenish the electron supply, there is an enzyme that splits water. Okay, it takes a water molecule, right, H2O, and breaks it apart into a hydrogen ion, actually 2 hydrogen ions, a molecule of oxygen, right, and that is where the oxygen that is released as waste comes from, and the electrons that were contained in the bond are then passed on to PS2 to replenish those missing electrons, right? The electrons that got excited and went into the electron transport chain. Okay, so we have our 2 high energy products, ATP and NADPH. Taking a look at what that actually looks like, we're putting it all together on the membrane. Here we see one set of the reactions embedded in the thylakoid membrane. Of course, if this were a real membrane, the entire surface would be covered with all of these proteins. And that's the reason for all of that surface area, to maximize the number of enzymes and photosystems that are contained. So a little bit about that electron transport chain there, right, between photosystem 2 and photosystem 1. Remember, they accept the high energy electrons and pass those electrons along using that energy. So they use that energy, right? The electron transport chain uses the energy from those electrons to pump hydrogen ions from the stroma, right, from outside the thylakoids, into the thylakoid space, right, inside those little stacks. So they are basically setting up a hydrogen ion gradient. Okay, and very important to remember, whenever there is a gradient, right, a difference in concentration across a membrane, there is energy contained within that gradient, right? The chloroplasts have invested energy to create that gradient, right? They use the energy from the electrons to pump the hydrogens across. So now that energy is still there within the gradient. So to get that energy out, we just the hydrogens need to flow back down the gradient, right? Diffuse back out of the thylakoid space. Now, they can't diffuse directly through the membrane, right? Because remember, membranes are impermeable to charged ions for the most part. So there is this large protein complex called ATP synthase. Okay, and that allows the hydrogens to flow down the gradient, right? In this case, out of the thylakoid space, back into the stroma. And as those hydrogens flow down the gradient, they are releasing the energy, and ATP synthase can capture that energy that's released from the gradient and convert ADP, adenosine diphosphate, to its high energy form of adenosine triphosphate, right? ATP. And that process is called chemiosmosis. Okay, and we will see this again. A very similar process occurs in cellular respiration to make the ATP molecules. Okay, so the electron transport chain creates ATP, but there's kind of a step in between where the energy from the electrons is used to create this hydrogen gradient, and then the energy from that hydrogen gradient then creates the ATP through ATP synthase. So before we move on to the next set of reactions, I do want to go and look at that energy and gradients again, because it is such an important concept. And like I said, we will see it again in respiration in part 3 of this video here. So looking at in terms of the chloroplast, right, the electron transport chain uses the energy from the high energy electrons to pump the hydrogen ions and create the gradient, right, going from the stroma into the thylakoid space, right? So the thylakoid space has a very high concentration of hydrogen ions. ATP synthase there, large protein structure, allows the hydrogens to diffuse back down the gradient, moving back out of the thylakoid, back to the stroma. And as the hydrogens flow down the gradient, they are releasing the energy, and that energy is then captured as ATP, right? ADP is converted to the high energy form of ATP. Okay, so those are the light dependent reactions, or sometimes you may hear them just referred to as the light reactions. Okay, remember location is thylakoids, major inputs, light energy, right, as our input of energy, and water. Remember, water is split to replenish the electrons in photosystem 2, and that's also the source of the waste product, oxygen, right? O2 gas is released to the atmosphere as the waste product. And then the two other outputs there, the two high energy products, ATP and NADPH, those then feed into the next set of reactions, which are the dark reactions or the Calvin cycle. Okay, I have them listed here as the light independent reactions. Okay, so these are now occurring out in the stroma. All right, so here we have the two high energy products there, ATP, NADPH, coming out of the light reactions and going in to drive the Calvin cycle. Right, and we see the low energy forms there, NADP plus and ADP, going back into the light reactions to be recharged. Right, and we see the other inputs and outputs there. I have the list of all of these things here, right? Location in the stroma, right? Out in the inside the chloroplast, but separate from the thylakoids. And the inputs, NADPH, ATP, right? Those are the energy molecules that were produced during the light-dependent reactions. And then our third input here is CO2, right? Carbon dioxide from the atmosphere as a source of carbon. And our main output that we're going to keep track of is this is a molecule called G3P, right? This is a 3-carbon organic molecule, glyceraldehyde 3-phosphate. And this molecule then forms the basis of all the organic molecules in the plant, right? Not just sugars, right? From this molecule, the plant's going to make all of its amino acids, all of its lipids, all of its nucleotides, right? All these organic molecules can be traced at some point back to this G3P, right? Because this is how carbon is brought in to organic molecules from the atmosphere. All right, so let's take a look at these reactions. And there's really one reaction here, or it's called the Calvin cycle. And it is a cycle because it runs in a circle, right? The first reaction, or I should say the last reaction of the cycle remakes the starting materials. So when we look at this, again, you certainly don't need to know the names of all of these molecules here, but we can kind of break the Calvin cycle down into three distinct phases, right? Where phase one here is called carbon fixation. Okay, and fixation is just a term that means attaching an atom to a larger molecule, right? So in this case, we are taking a carbon atom, right, from CO2 from the atmosphere and attaching it to a larger organic molecule, right? And you see the starting material there on the right is that 5 carbon molecule, goes through this initial reaction, and now there's a 6 carbon molecule, right? So we have increased the number of carbons in our organic molecule by 1. And the enzyme that performs this reaction is called Rubisco. Okay, and that enzyme is probably the one that you may need to know, and we'll come back to it in a little bit at the end when we talk about some details with the Calvin cycle here. Okay, so that's phase 1, carbon fixation. Phase 2 is this reduction phase, where it's just kind of rearranging all of these carbon atoms, and you see the input of the energy there, right? ATP and NADPH are both being used. Remember, those were produced in the light-dependent reactions, right? So here's where those energy molecules are being used up, right? ATP goes to its low energy form, ADP, NADPH goes to its low energy form of NADP+. And at the end of this phase 2, our final product is released, right? G3P, this sugar, right? And you see there that this then goes on to very easily form glucose, which many people kind of treat as the final product of photosynthesis, but that's, one step away. We need to take 2 G3P molecules, 2 of these 3 carbon molecules, and attach them together to get the 6 carbon glucose, right? And also all the other organic molecules in the plant also come from this G3P. Okay, and then finally we have phase 3 of the Calvin cycle, and that is the regeneration of that starting molecule, right? And this basically just completes the cycle and gets us back to the beginning. Okay, and as I said, I want to talk a little bit about Rubisco, because not only is it a very important enzyme in terms of fixing carbon, it's also fairly unique. In terms of enzymes, we usually think of them being very, very specific for their substrate and only working for one particular set of reactants. Rubisco, though, is a little bit different, because it will also use, as a starting material, oxygen instead of CO2. Now, that's a problem because the whole point of this initial reaction here is to add carbon to our 5 carbon molecule. Oxygen cannot function as a source of carbon, right? There are no carbon atoms there. So Rubisco, if it uses oxygen, that is actually a wasteful process. And there are a few alternate pathways of carbon fixation that plants have evolved, that plants have adapted to avoid this wasteful process that is called photorespiration. Okay, so Rubisco is here in the middle. Oh, let me go to the picture. There we go. So there's Rubisco in the middle, right? If it uses carbon dioxide, that is photosynthesis, right? And everybody's happy. We're using the energy, and the plant is producing these organic molecules, right? The sugars. In photorespiration, right? If oxygen comes in and Rubisco uses that, then there's no additional carbon added to the cycle, but the cycle will still spin, and those high energy molecules are used up, but the plant doesn't gain anything, right? It didn't make any usable outputs. So this photorespiration, right? Rubisco catalyzing a reaction with oxygen is very wasteful. So plants have evolved ways to avoid this photorespiration from taking over. So when would this photorespiration happen? Right. So on the bottom of a leaf, there are these pores that are called stomata. Okay, and they are what will normally let the gases in and out of the leaf. Right. You see there the CO2 coming in and the oxygen coming out. But what will also be coming out of these stomata is water, right? When the water moves up through the stem or the trunk up into the leaves, right, that water is going to then escape through the stomata. So plants, one of the adaptations they made to move onto dry land is these stomata can open and close to conserve their water. So on very hot, dry conditions, the plant is going to close all of its stomata, as shown there on the right, and keep all of the water inside. Now, the problem with that, it's also blocking the gases from exchanging, right? So carbon dioxide cannot get in, right? And if you think, a hot, dry day, that's probably also going to be very sunny. The light-dependent reactions are going to be running, creating oxygen. And that oxygen, that waste product, now cannot get out of the leaves. So these air spaces here in the spongy mesophyll layer are going to fill up with oxygen. And that is bad for the plant because now photorespiration is going to start to take over. So these plants that live in hot and dry conditions, right, have evolved these what are called alternate pathways to prevent the Rubisco enzyme from being exposed to these high concentrations of oxygen. Okay, and there are two sort of alternate pathways, right? Two adaptations that plants have made in order to prevent photorespiration. Okay, and they are C4 plants and CAM plants. Okay, so C4 plants move these steps of photosynthesis into two separate cells. Okay, in the mesophyll cell, and that's the one that is exposed to the air spaces, right? The CO2 is going to come in, and that will be converted into an organic acid. Okay, and the reason it does that is because CO2 as a dissolved gas can't really be stored. Right? The plant cell can't store up large amounts of CO2, but converting it into this organic acid, that can be built up. Right? The plant can store high concentrations of this organic acid. So this organic acid made in the mesophyll cells is then moved into a different cell called the bundle sheath cell that is a little bit deeper inside the leaf. Okay, and this bundle sheath cell is not exposed to those air spaces inside the leaf. So it's not going to be exposed to those high levels of oxygen that are going to be built up while the stomata are closed to conserve water. And these bundle sheath cells are where Rubisco is found, right? And the Calvin cycle occurs there. As the organic acid is moved in from those mesophyll cells, the organic acid is broken back down, CO2 is released, and the Calvin cycle runs as normal without Rubisco seeing that high concentration of oxygen. Okay, in the other hot and dry adapted plants that we call CAM plants, they separate the processes out by time. So at night, stomata are open, right? Generally cooler. Water loss is not as large of a problem. So again, the CO2 is brought in and it's converted to an organic acid, right? The plant can store up large amounts of this organic acid. Then during the day, when the light reactions are running and creating the ATP and NADPH needed for the Calvin cycle, that those organic acid molecules are broken back down, the CO2 is released, and the Calvin cycle can run as normal without Rubisco seeing that high concentration of oxygen. Okay, so these alternate pathways are very important for plants that have adapted to these hot and dry conditions so that they can close their stomata, conserve their water, but not undergo that wasteful process of photorespiration, right? Which is just using Rubisco using oxygen instead of carbon dioxide and not creating any new organic molecules. All right, so that is the end of our endergonic pathways, right? Our energy capturing pathways. And in the final part of this video, we will get into the energy releasing exergonic reactions of cellular respiration. So the final set of reactions that I will go over here are the energy releasing pathways of cellular respiration. Okay, so since these are the energy releasing pathways, of course, that means that these will all be exergonic reactions, meaning that the end products are at a lower energy level. So even though we think of this as creating the cellular energy molecule, the end product, all of the ATP, will be at a slightly lower energy level than all of the input starting organic molecules. Okay, so that is very important to remember, that along with every other energetic transformation, there is a little bit of energy loss. Okay, so starting just looking at a broad overview of cellular respiration, one thing that is very important to remember is the way that photosynthesis and respiration work together in this overall cycle of moving carbon through living things, right? And through an ecosystem, right? Something that we will definitely talk about in the final ecology unit 8 review. But when we look at this, when we look at these two processes in this overall process, right, we have energy coming in to the ecosystem, mainly from sunlight, and that is captured by the chloroplasts through photosynthesis, and that takes the carbon dioxide and puts it into the organic molecules. And then cellular respiration in the mitochondria takes those organic molecules, breaks them down, and that energy that is released some of it is captured as ATP, and then the carbon is then released back into the atmosphere as CO2 and can then enter back into photosynthesis, right, forming this big cycle of carbon atoms moving through the ecosystem and being recycled. So looking at respiration itself, here are the different stages that we will talk about. Right? I'll start with glycolysis and then move through the reactions that occur in the mitochondrion with the citric acid cycle or Krebs cycle, and then the oxidative phosphorylation. All of these processes occur in multiple steps. And remember, this is something that I talked about way back in part 1 with the stepwise release of energy. If the cells instead released all of the energy at once, then it would be, as it says there, it would be explosive, right? And all that energy released at once, very hard to capture any of that released energy in a useful manner. When the energy is released step by step through these multi-step metabolic pathways, it's much easier for the cell to capture more of that energy and maximize the amount of ATP that is being produced, right? And that, remember, will be the final goal, the ultimate goal of all of these cellular respiration pathways, is to make as much ATP as possible from the breakdown of these starting organic molecules. before we get into the pathways and the reactions, just real quickly want to talk about some of the structures, right? And that, of course, is the mitochondrion, right? This is the organelle for cellular respiration. And structurally, we see it is just like the chloroplast. There is a double membrane, right? The outer membrane surrounding the inner membrane. Though in this case, the inner membrane will become important. There are some reactions that occur there. And structurally, we see that the inner membrane is all folded up, right? Again, whenever we see folds like that, that means increased surface area. Right? So something is happening here where the mitochondrion needs all of that surface area. And then the inside of the mitochondrion, right, inside the inner membrane, is called the matrix, and we'll talk about the set of reactions that occurs there. And then the space in between the membranes, right, in between the inner and the outer membranes, is referred to as the intermembrane space, and that will also become important, right? So a few more structures here, or a few more regions that are important than what we saw in the chloroplast. The first set of reactions that I will talk about here is glycolysis. Okay, and just like with photosynthesis, right, the important stuff up here, right, location, inputs, and outputs. Right, so for glycolysis, the location is the cytosol, right? So this is happening just in the cell, and that is important because every cell, every domain of life can perform glycolysis. So remember that things like bacteria and archaea that don't have any of these internal organelles still perform glycolysis, right? Because all the enzymes are just located in the cytoplasm. The major input here is glucose. Certainly that is not the only energy source that cells can use, but we do tend to talk about it and use it as a starting point because every cell can use it, and it makes it a very nice and easy place to start. And then we have the outputs there, 2 ATP molecules, 2 NADH molecules. Those are both high energy products. And then 2 pyruvate molecules. And notice I have the little star there after pyruvate, just to note that that is where most of the energy is contained at this point. Okay, so now as we go through respiration, it's important to not only keep track of outputs in terms of high energy and waste products, but it's important to kind of keep track of where the majority of the energy is contained. Right, so at the end of glycolysis, made a little bit of ATP. Remember, that is the cell's main goal here. Some NADH, this other energy product, but most of the energy from that starting glucose is contained within the 2 pyruvate molecules. So looking at the reactions of glycolysis, again, I'm not even going to show all of the steps because they are not important. What is important though are these 2 different phases of glycolysis. Right, the initial energy investment phase there, where glucose comes in. Remember, that is the starting material, and the cell actually makes this initial investment of 2 ATP molecules. Right, there's a little bit of energy put in to get the process going. Right, and those 2 ATP are converted to the lower energy form of 2 ADP. And then those products then enter into the energy payoff phase, where the cell now gains some energy molecules, right? 4 ATP are formed in this energy payoff phase, and 2 NADH, right? And those the NADH molecules are high energy electron carriers, right? Very similar to the NADPH molecule that we saw in photosynthesis, right? NADH functions in respiration. And then, of course, the 2 pyruvate molecules that are formed. Those are 3 carbon molecules, and they come from the glucose molecule literally just being broken in half, right? That's basically what is happening here, right? The 6 carbon glucose is broken in half step by step, and forming these two pyruvate molecules, and the energy that is released from that breaking is captured as ATP and NADH. I'm not sure how important all these numbers of molecules are going to be. Sometimes the College Board asks about them, sometimes they don't. I have them here just so that you can see them. The net gain then of ATP, remember there was 2 initially put in the engine investment phase, right? 4 came out at the end. So our net gain so far is 2 ATP molecules per glucose. The formation of the ATP molecules here during glycolysis is a process called substrate level phosphorylation. Okay, and this is a little different from the way that ATP was made in photosynthesis and the way that we'll see ATP made at the very end of respiration when the bulk of the ATP is produced. So substrate level phosphorylation basically means that there is an enzyme that binds ADP, right, adenosine diphosphate, and some other organic molecule that's just labeled substrate here that has a phosphate group. And this enzyme pulls that phosphate off the other organic molecule and attaches it to ADP. Right? So there is some organic molecule, some substrate that this phosphate group is attached to initially before it is made into ATP. So that's why it's labeled substrate level phosphorylation. Okay? When we get to the end of respiration, we'll see that the ATP there is made a little bit differently. Okay, so following the energy, remember most of the energy at the end of glycolysis was contained in those 2 pyruvate molecules. So those 2 pyruvate molecules are now going to move into the mitochondrion and undergo the next set of reactions, the Krebs cycle or the citric acid cycle, right? 2 different names for the same process. Okay, so location, mitochondrial matrix, right? So this is the inside of the matrix, right? All the way inside through both membranes. The input is pyruvate, though technically for talking about the Krebs cycle, the input is this molecule called acetyl coenzyme A, which we'll talk about in a second. And there we see our outputs, right? A little bit of ATP is produced, and NADH and FADH2. Okay, most of the energy here is in those 2 electron carrier molecules, right? NADH and FADH2. Okay, the structures of those molecules and what those names actually stand for, not terribly important, but the function is important. Right? These are the high energy electron carriers, right? And we'll see them coming back in the next set of reactions. One final product here of the Krebs cycle is carbon dioxide, right? And that is released as a waste product. Okay, so the steps here of the Krebs cycle, first there is this conversion of the pyruvate from glycolysis into this other molecule, this 2-carbon molecule called acetyl coenzyme A. Okay, so this happens right after it's brought into the mitochondrion, right? We see it there going through the 2 membranes into the mitochondrial matrix. Pyruvate is broken down. One of the carbons is stripped off, and that is released as a waste carbon dioxide. And now we have this 2-carbon molecule called acetyl coenzyme A, right? And this is what then goes into the actual Krebs cycle or citric acid cycle, right? Like I said before, 2 names for the same thing. Okay, and you notice here I don't even have all of the molecules listed there or any of the enzymes because those are not important to know. What is important is, as we went over at the beginning, right, location. This is happening in the mitochondrial matrix. The input there, you see there at the top, acetyl-CoA is coming in, and the products, right? The high energy products, certainly ATP, right? A little bit of that is made. And then we see NADH and FADH2, right? Those are the high energy electron carriers, right? And that is where those two molecules are where most of the energy is found after the Krebs cycle, right? And we also see the waste product being formed over there, the two carbon dioxide molecules, right? And those are going to be released, right? In animals like us, those would be exhaled, right? That's the Krebs cycle, right? So at this point, we've made a little bit of ATP, but most of the energy is in those high energy electron carriers, NADH and FADH2. And remember, there's also a little bit of that of NADH made during glycolysis. So the final step here will be getting the energy out of those electron carriers, right? And that is the electron transport chain and formation of ATP through chemiosmosis. Okay, so this occurs on the inner mitochondrial membrane, right? Remember that had all of those nice folds, right? All of that great surface area for all of these reactions to take place. The inputs there, NADH, FADH2, right? And those are coming from glycolysis and from the Krebs cycle. And finally, our outputs, ATP, right? We're going to make a lot of ATP, so that is where most of the energy is going to be found. And there is some more waste, right? CO2 is released as a gas, and there is some water also produced as a waste product here. So looking at these reactions of the electron transport chain, very similar to what we saw in photosynthesis with that electron transport chain. But here, the high energy electrons are coming from these carrier molecules, right? NADH and FADH2. So those high energy electrons are donated from those carrier molecules to the beginning of the transport chain here. And the electrons are passed from protein to protein, right? Again, the names of all of these proteins in the chain are not terribly important. And as those high energy electrons are passed along, the energy is used to pump hydrogens, right? And here the gradient is going from a low hydrogen concentration in the matrix to a higher concentration out in the intermembrane space. Okay, and remember, whenever there's a gradient, there is energy within that gradient, right? The cell used energy to create the gradient, so that energy is still going to be there. Okay, at the very end of the electron transport chain, the final electron acceptor is oxygen, right? And this is why oxygen is needed for this aerobic respiration, right? It sits there at the very end. Remember, oxygen is highly electronegative, so you can kind of think of it as oxygen is pulling the electrons down the chain, and at the very end there, it's accepting those electrons and mixing or reacting with some of the hydrogens to form water, right? And that is the waste product. Okay, the ATP from respiration, right, is produced through chemiosmosis, and that is occurring, again, very similar to what we saw in photosynthesis, right? There is this large protein complex, ATP synthase, and that is going to allow the hydrogen ions to diffuse down the gradient, right? And that releases the energy, and that ATP synthase can capture that released energy from the gradient as ATP, right? So it uses that energy to link ADP and the inorganic phosphates, the phosphate groups that are just floating around inside the mitochondria, to make ATP. Okay, so that is different then from the substrate level phosphorylation that we saw in glycolysis and that also occurs in the Krebs cycle, right? This is this oxidative phosphorylation, okay? And it's driven, again, by this energy from the hydrogen gradient that was produced by the electron transport chain that got the energy from those high energy electron carriers. Okay, so that's kind of tracing the energy all the way back. Looking at all of this together, and again, I'm not sure how important the numbers are going to be, but we get an idea here of where the energy is made. Right, so starting with glycolysis out there in the cytoplasm, we get a couple, 2 ATP there, 2 NADH. Most of the energy is then going on into pyruvate oxidation and the citric acid cycle. Right, that then produces more NADH and FADH2, and those drive the oxidative phosphorylation, the electron transport chain, and chemiosmosis, and that is where, as you can see, the bulk of the ATP is produced. Okay, so overall, we get somewhere between 30 and 32 molecules of ATP per glucose molecule. Okay, the cell doesn't always get that much, right, because there's a lot of different things that can happen here. Some of these intermediates can go off and be used elsewhere. So that number there is just really, as it says, a maximum amount that the mitochondrion could produce under perfect conditions. All of this here, or at least the part that's in the mitochondrion, is referred to as aerobic respiration, right? Meaning that it requires the input of oxygen, right? Remember, oxygen sits as that final electron acceptor at the end of the electron transport chain. But not all organisms are aerobic, right? There are some organisms that are strictly anaerobic, right? Meaning that they do not use oxygen, or that oxygen is actually toxic to them. So if there is no oxygen present, or if it's an organism that just hasn't evolved this process to use oxygen, then there are other pathways called the fermentation pathways, right? And this is a way for the cells to continue to get energy without the use of oxygen. So when we look at these fermentation pathways, these anaerobic pathways, the pyruvate molecule really functions as this branch point here, right? So glucose is coming in, glycolysis occurs in the cytoplasm, right? Glycolysis is anaerobic, right? There is no oxygen required. If oxygen is present, then the pyruvate is moved into the mitochondrion for the processes that we just talked about. If there is no oxygen present, right? If the cell is in an anaerobic state, then the pyruvate is moved into one of the anaerobic fermentation pathways. Okay, and those two fermentation pathways are the alcohol fermentation or the lactic acid fermentation. Okay, and in both of these, there is no additional ATP made in the fermentation side, right? So fermentation would be along the bottom there, right? Going from pyruvate. In the case of alcohol fermentation, the pyruvate is broken down into a 2-carbon alcohol, ethanol, and then in lactic acid fermentation, the pyruvate is converted into a different 3-carbon molecule called lactate. So there's no additional ATP made. What is the point, right? Why do these pathways exist? Well, the reason is that glycolysis is an energy producing pathway, right? It does create ATP, but it also uses up this NAD+, right? The precursor, the low energy form of NADH. And without oxygen, remember, there's no way to get the energy out of the NADH, right? That has to go into the electron transport chain. So what glycolysis does is it takes that NADH and converts it back into NAD+. And that NAD plus then allows glycolysis to keep moving. Right? If this fermentation pathway didn't exist, right, glycolysis would run, the NAD plus would all be converted to NADH, and then the cell couldn't do anything with that, right? Because under anaerobic conditions, the NADH cannot be broken down to harvest the energy and make ATP. Okay, so fermentation pathways, though they do not create any new ATP themselves, they are very important in keeping the glycolysis pathways going. And for many microorganisms, these glycolysis pathways, these anaerobic pathways, provide plenty of ATP to keep the organism alive. Okay, so that is the end of our Unit 3 energy review. I hope that all made sense, and good luck on your AP.
Dropping science like Galileo dropped the orange.